Welcome back to Dr. Liu's VCE Biology. Today's topic is regulation of biochemical pathways, as presented in the 2025 VCE Biology exam. Let's learn together. If teachers, parents, or students notice mistakes in my explanations, please provide me with your feedback. A. Incorrect. Between 0 degrees Celsius and 30 degrees Celsius, the activity is 100%. If the tertiary structure were unraveled, the protein would lose its function and the activity would drop. B. Incorrect. The primary structure, the sequence of amino acids, is held together by strong covalent peptide bonds. These bonds are generally not broken by changes in temperature. Denaturation affects the higher order folding, not the sequence itself. C. Correct. Haemoglobin is a protein with quaternary structure, made of 4 polypeptide subunits, as seen in the provided diagram. The graph shows that within this temperature range, the protein loses all activity, which is the hallmark of denaturation. D. Incorrect. At 40 degrees Celsius and above, the protein has denatured. Denaturation involves the disruption of the secondary, tertiary, and quaternary structures. Therefore, the secondary structure is definitely affected, changed, lost. A. Incorrect. The inhibitor binds to the active site of the enzyme, ATP synthase, not to the substrate, ATP, ADP, itself. B. Incorrect. By blocking the enzyme, the inhibitor prevents ADP from being converted into ATP. This would actually lead to an increase in the number of ADP molecules, not a reduction. C. Incorrect. Binding to an allosteric site is the definition of non-competitive, or allosteric, inhibition. Competitive inhibitors specifically target the active site. D. Correct. Because ATP synthase is responsible for phosphorylating ADP into ATP, inhibiting the enzyme means the recycling of ADP back into ATP stops. Consequently, as the cell continues to use up its existing ATP for energy, the concentration of ADP molecules will initially rise, because they cannot be processed by the inhibited enzyme. Mark 1. Enzymes catalyze biochemical reactions by lowering the activation energy required for the reaction to proceed. Mark 2. They do this by providing an active site that is complementary in shape to a specific substrate. When the substrate binds, it forms an enzyme-substrate complex, which destabilizes chemical bonds and allows the reaction to occur more rapidly. The graph below shows the activity of the enzymes coded by the genes within the TRP operon. At 20 minutes, the bacteria were shifted from an environment containing TRP to an environment without TRP. The solid line represents a model simulation of enzyme activity, while the triangles and squares represent 2 sets of experimental data. To decode the stem of this specific 5-mark question, students should follow a 3-step mental process before they even start writing. Step 1. Decode the story in the graph. Before looking at the mechanisms, students must interpret the data. The baseline, 0 to 20 minutes. The activity is at 0. Student logic. Why is it 0? Because the stem says there is tryptophan present. The cell is being lazy and saving energy by not making what it already has. The trigger, 20 minutes. The environment shifts to without TRP. Student logic, the supply is gone. The cell needs to flip the switch on immediately. The plateauer, 60-minute plus. The activity levels off. Student logic, the cell has made enough enzymes to reach a balance. It's not overproducing, it's steady. Step 2. Identify the tools, the mechanisms. The question explicitly asks for repression and attenuation. Students should view these as two different brakes on a car. Repression equals the handbrake. It stops the car from moving at all. It works at the very beginning, transcription initiation. Attenuation equals the emergency brake. Even if the car starts rolling, leaky transcription, this brake can stop at midhill, transcription elongation. Step 3. Link tools to the story. Now, the student connects the biological facts to the graph they just analyzed. This is where they work hand in glove. The explanation, the how to explain 0 to 20 minutes, both brakes are on. Repression, high TRP equals active repressor equals blocked operator. Attenuation, high TRP equals fast ribosome equals 3 to 4 terminator loop forms. Result, no mRNA made, 0 enzyme activity. To explain the shift, 20 to 60 minutes both brakes are released. Repressor falls off, ribosome stalls at TRP codons, forming the 2 to 3 anti-terminator. RNA polymerase is now free to fly through the whole operon. To explain the steady state, 60 minutes plus the fine-tuning. This is the working together part. As enzymes make tryptophan, the levels slowly rise. The cell doesn't want to overshoot. Repression and attenuation start to flicker on and off to maintain that perfect steady line on the graph. (0 to 20 minutes), high TRP, repression and attenuation. Repression, TRP, co-repressor, activates the repressor to bind the operator, blocking RNA polymerase. Attenuation, rapid translation of leader peptide forms a 3 to 4 terminator loop, stopping transcription. 20 minutes, TRP-free, activation, repressor detaches and ribosome stall, forming a 2 to 3 anti-terminator loop, allowing RNA polymerase to transcribe structural genes. 60+ minute, steady state, synergy. Hand-in-glove regulation, both mechanisms work in tandem, as a negative feedback loop. They fine-tune expression to match the cell's metabolic demand, maintaining homeostasis, and preventing resource waste. Exam strategy, the mark-earner checklist. To save time, students should focus on these marks per word winners, operator, use this instead of the gene or promoter. Transcription, clarify that this, not translation, is what's being stopped. Terminator versus anti-terminator, use these specific names for the loops. Structural genes, mentioning that these are the final goal of the operon shows deep understanding. The exam mode mental process. Identify the target. The question asks for limitations of the model, the line, not the experiment, the dots. Look for the gap. Where is the line most different from the dots? Between 20 to 40 minutes. Diagnosis. The dots are faster and messier than the line. The model is too smooth and slow. Limitation 1. The model is oversimplified and fails to account for random biological variations, a stochastic noise, in tryptophan levels within a living cell. Limitation 2. The model assumes idealized kinetics, underestimating the speed at which the repressor detaches, or the anti-terminator loop forms in response to a sudden environment change. Why this gets the marks? Oversimplified, this is the gold keyword for any model-related question. Random biological variations directly addresses why the dots aren't perfectly on the line. Underestimating speed explains the specific gap shown on the graph at the 20-minute mark. Critical red pen warning. Do not write. The scientists might have made a mistake in the experiment. The examiner says, this is the most common reason students get 0, 2. Always blame the math simulation, not the measurement. The core stem, the what? The change. A mutation has reassigned the UGG codon. Shift, it no longer acts as a stop codon, signals termination, but now codes for the amino acid tryptophan, TRP. The logic chain, the how. To provide a complete justification, the student must follow the domino effect of protein synthesis, DNA to translation. The ribosome ignores the previous finish line. Primary structure. Additional amino acids are added, lengthening the polypeptide chain. Tertiary structure. The new sequence causes the protein to fold differently. Biological fitness. The double-edged sword. The new shape either breaks the protein or enables new regulatory functions like attenuation. Exam-style response. The molecular change. 1 mark, because UGG no longer signals translation termination, the ribosome continues adding amino acids, resulting in a longer primary structure. The biological outcome. 1 mark, this altered sequence causes the protein to fold differently, changing its tertiary structure, leading to a gain or loss of function. This impacts metabolic efficiency or regulatory sensing, like attenuation, ultimately increasing or decreasing bacterial survival. 1. Enzyme dynamics and inhibition. Catalysis. Enzymes lower activation energy via the induced fit model. Denaturation. High temperatures disrupt tertiary or quaternary structures. Primary structure, peptide bonds, remains intact. Competitive inhibition. Inhibitors block the active site. If an enzyme is inhibited, substrate levels, e.g., ADP, initially rise. 2. The TRP operon. Hand-in-glove regulation repression. The on-and-off switch. High tryptophan levels activate a repressor to bind the operator, physically blocking RNA polymerase. Attenuation. The dimmer switch. High tryptophan levels lead to a 3 to 4 terminator loop, stopping transcription prematurely. 3. Model versus reality limitations. Computer models are often oversimplified, ignoring random biological noise variations, and assuming idealized kinetics that don't match real-world speed. 4. The double-edged sword of mutation. Molecular change, UGG, stop to TRP codon, leads to no termination, resulting in a longer primary structure. Biological outcome. Altered sequences cause different folding, tertiary structure, leading to a gain or loss of function that ultimately increases or decreases bacterial survival. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.